yeah, I'm really excited to be here talking about the 3D Moss project and to have a chance to talk to you a bit about my contribution to this larger investigation into Syntrichia. And I'm going to be, as Brent said, picking up from Mel's presentation and kind of seeing how that can connect all the way to ecology and the natural habitat and interaction with environment, specifically looking at UV and desiccation tolerance. All right, so we talked about, well, this whole workshop is about this dryland moss group, and drylands can come in many flavors, but deserts are among them, kind of the more arid drylands. And this is where my work takes place in the Mojave Desert, and specifically in this UC Natural Reserve within the Mojave National Preserve. And I focused on Syntrichia cananervis because it's just awesome, as you'll see more of. And here's a little video. Brent showed you a great re-wetting video, and you'll see a lot more with Javi's talk later. But I want to stress that I added some water here, and this is not sped up. This is real time. And as you can see, it gets some water and opens up and turns green before your eyes. And then dries up again when the water is gone. And I find this process intriguing and exciting. And in thinking about their habitat, I always get stuck here. Okay, where they're dry for most of the year because it doesn't rain very much in the summer. My real question and curiosity comes down to, how does it do it? How does it survive? We know it has this amazing desiccation tolerance, but it's still alive even while it's dry, and still gets hit with sun in the desert. And I know from spending just a few hours in the desert sun that it's really powerful and damaging. And so I wanted to focus on that for my research. So a bit about light and what plants use for photosynthesis. These are absorption curves, and where they're tall, it shows where it's most absorbed and therefore most important. So the chlorophylls absorb largely blue and red light, and these carotenoids have a little bit wider spectrum of absorption, and maybe even into the UV range, which is over here. These are all pigments, and we'll talk more about those. UV light in general is thought to be damaging. It can certainly be damaging, but there's increasing evidence that it plays regulatory roles too. And so that's something to keep in mind with these results. So Brent gave an introduction to these, so I can breeze through this, but transcriptomics and metabolomics are, what I'm talking about today. And transcriptomics is a part of this, what's known as the central dogma of biology. The DNA, which Mel talked a lot about, is all of the coding equipment for the whole plant. And while RNA is sort of transcribed when needed. So if we look at the RNA, it can give us a clue about what the plant needs in that moment. So transcriptomics is all about identifying and counting those RNA transcripts to get an idea of what the plant is doing physiologically. Then metabolomics comes later, after proteins are formed from the RNA. Proteins can be enzymes and work to make all kinds of different molecules, including sugars, hormones, pigments that we talked about. And so any molecule that's kind of part of the plant's life could be considered a metabolite. And metabolomics is all about identifying and quantifying those. So together, if we look at both of these, we can really get some insight into how an organism is functioning. And if you set up an experiment, you can see how an organism is responding to their environment or a particular stimulus you give it. So that is what I did. I set up 2 experiments. I'm going to tell you about a little piece of each of them. One is a UV removal experiment from the natural habitat in that Mojave Desert site. And the other is a controlled laboratory experiment looking at UV and desiccation. This first experiment in the natural habitat looked like this, as Brent showed. This is the ground there, and all of this dark stuff is Syntrichia cananervis. There's some other mosses here, and probably lichens, certainly cyanobacteria, all organisms that Matt will talk to you about later. But I'm looking at the Syntrichia cananervis, and I set up these windows that either block or filter UV or transmit it as a control. So the only thing different between these should be the UV, whether it has it or not, and also collected some what I call site reference, which were just completely free and wild plants. There are unmanipulated, no windows. There were 20 of these triplets, and I left them there for a year. Now, after collecting them, the first thing I wanted to do was just take a look. And the unmanipulated plants looked pretty typical for Syntrichia cananervis, at least in the wild. This dark pigmentation and kind of rust colored on the bottom. And the UV transmitting windows, which have UV allowed to go through, look pretty similar. Although to my eye, they look a little paler sometimes, but not a big difference. But the UV filtered windows have a huge difference in that the plants are very, very green and seem to lack this dark pigmentation. And this is pretty atypical for this species. You might find them this green if it's growing in a really shady spot, but not out in the sun in the desert. So that was pretty exciting. This first finding that UV appears to cause a suntan effect, just like it does in humans. But that green color made me pretty curious about the pigments in it. And so I looked at some photosynthesis related pigment metabolites, including chlorophylls and zeaxanthin, which is a carotenoid, like I mentioned. And what I found, the blue here is the UV filtered, and we're comparing that to the orange, which is the UV transmitted window. And in both this chlorophyll ratio and zeaxanthin, it's higher when UV is filtered out. And that's interesting because both of these, when it goes up, is a signal that it's a plant in a high light environment. And we in fact took some of the sunlight away, took the UV radiation, and yet they're responding as if it was sunnier. So that was a first surprise and started to give us clues about what was happening. But I wanted to note that although I mentioned zeaxanthin is a pigment, it also is an antioxidant, and as other carotenoids are, and it's one that you may even be familiar with as a supplement people take for eye health, and so a lot like beta carotene and other carotenoids. So an antioxidant went up when we took UV away, and that was a surprising finding. So I looked at the transcriptome too, and found a similar pattern that here in the blue, when UV was taken away, antioxidants associated transcripts went up with UV removal, which is opposite of what I would have expected, because typically UV causes oxidative stress. So again, taking UV away, and they're responding as if it's kind of a stressful event. There were also these flavonoid-related transcripts that went up with UV removal. Now, flavonoids are also antioxidants, or can be, but they're also known to be sunscreen pigments. But the interesting and tricky thing about these transcripts is they actually break down flavonoids. And so when the transcripts go up, you get less flavonoid metabolites. And so these going up is thought to be a decrease in flavonoids, which might be that less dark coloration we were seeing in the plants when UV was removed. All right, so I also did this controlled laboratory experiment where I had these two UV environments, low UV and high UV. And then I also kind of crossed that with a desiccation treatment. So in each case, they were either hydrated or desiccated. And I grew them in a growth chamber in the lab. Now doing this controlled environment allows me to kind of zoom in even more on these particular stresses, UV and desiccation, whereas the field one was kind of the whole environment at once. So looking at a few metabolites that increased as UV went up, the bright blue is the high UV, and this is in the hydrated treatment and desiccated. You can see this alloglu and D-xylose especially go up a lot with UV and aren't changed a ton with desiccation, but maybe go up a little bit in this case and down here. The interesting thing about these are alanine and glutamate are amino acids, and this D-xylose is a sugar. And both of these are thought to be important for desiccation tolerance. And so this is interesting. Desiccation didn't increase these, UV did. And so it might be a case of what's known as cross tolerance. UV induces production of a metabolite that's useful for a different stress, desiccation. And finally, more transcripts from that laboratory experiment. There were two ELIPs, early light inducible proteins that Mel talked about, that were increased with desiccation, just as he found in the genome study, and maybe in some cases with UV, increasing UV as well. And so it's possible that ELIPs are involved in UV tolerance and obviously in desiccation tolerance. So another case of possible cross tolerance between these two stresses. All right, so what did we learn? This was a whirlwind of some of my results, but in wild Syntrichia cananervis, UV causes a suntan, we know that, and it might be a flavonoid pigment. It alters photosynthetic pigments, chlorophylls and carotenoids. And removal increases stress-related transcripts, such as antioxidant transcripts, as well as antioxidant metabolites. In a controlled setting, we found evidence of overlap in UV and desiccation response. I just want to shout out all the funding sources. Obviously, the 3D Moss project funded a lot of this work, but I had many other funders that I'm really thankful for because it truly wouldn't have happened without them. Also, my collaborators, many of whom are in this group, and a lot of student researchers who have contributed to this project. So yeah, thank you all. I'll be looking in the chat if there's any questions that my colleagues did not answer. Thanks.